So our next discussion is how evolution works, and the theory is, misconception is that organisms evolve during their lifetimes, and that is not true. Okay, this discussion is going to relate to the evolution of populations. Okay, an individual can die because it's missing a limb or an eye or something like that, but the populations evolve. Okay, so we're going to look at genetic variations in populations. This is also called, well, we were talking about population genetics and evolution. And there's a lot here. I'm going to kind of go pretty fast through some of these slides. So we're really talking here about microevolution, how a population can change, not a whole species. That's macroevolution, and that's for the next discussion. Okay. So we're looking at how populations change genetically over time, and then we're putting it with our knowledge of genetics with the theory of evolution. Okay, and so this just quick goes through a couple definitions. Okay, the gene pool is all of the genes in a population at any one time. Okay, so we're talking about the population of this classroom, all of the genes each of you has. Okay, that's the gene pool. And so we can look at the gene pool in this herd in Alaska, or in this herd in Alaska, or in this herd in Alaska. It's a gene pool. Okay? The first thing we need to understand before we get into this is something called the Hardy-Weinberg Theorem. The Hardy-Weinberg Theorem says that frequencies of genes will, in a population, will remain the same from generation to generation if there's no natural selection. Now, we're going to talk about Hardy-Weinberg in some great gory detail. Okay? But, so we're not going to go through that. Well, we can just glance at this a second. So if we cross a red flower and a white flower, F1 is all pink. When the pink flowers reproduce, they're going to get half red, half pink, and half white. But what hasn't happened is we haven't had a change in the number. We haven't had any change in how many alleles we have. For example, this one has 2 red alleles. This one has 2 white alleles. Its offspring has 50% red, 50% white. Down here we have 50% red, 50% white. So we've not, even though the colors change because of incomplete dominance, the frequency of the alleles has not changed, how many of them we have. So, in a population where gametes contribute to the next generation randomly, allele frequencies will not change. You don't necessarily have to write that down. Hardy-Weinberg Equilibrium describes a population in which random mating occurs. Now, I'm going to skim through a couple slides here because population genetics, or Hardy-Weinberg, we're going to talk about in class in great detail. There are 5 conditions for Hardy-Weinberg to be met, and you need to know these. Okay, so in order for populations to not change, we have to have an extremely large population. We have to have no gene flow. We have to have no mutations. We have to have no natural selection. And we have to have random mating. Okay, the 5 conditions, you need to know these, so stop this and write it down, are you have to have a large population, no gene flow, no mutations, random mating, and no natural selection. So now I'm going to kind of provide you with a list of things that cause populations to change. Okay, and we're not going to do a whole lot of description of each of these. We're going to provide you with a list of things that cause populations to change. Process number 1 is mutation. Okay, process number 2 is sexual recombination. Okay, and I'll take a minute to talk about each of those, and then we'll go on to a couple others. Okay, mutations, remember, are the change in the sequence of DNA. They cause new genes and alleles to arise. The difference between black hair and blonde hair is whatever the parent had. Think about this. Whatever the very first person had, anything else is a difference, right? That would be a mutation from their original DNA. There must be a different letter in the sequence. If the sequence for brown hair is ACTG, the sequence for black hair might be CCTG, and I'm just making that up. And a point mutation is a change in one base in the gene. We have chromosomal mutations, which we talked about before. And mutation rates aren't very high, but average about one mutation every 100,000 genes per generation, and we only have 33,000 genes in us, so that's not very high. But if we have enough generations, there's a chance. Sexual recombination, what is that? Well, let me go back a second. That'd be things like crossing over, okay, where we get the switching of genes. That'd be things like you are not randomly mating, actually. A dog does not randomly mate. It mates with another member of its kind. It mates with another German Shepherd, or we make them mate with other German Shepherds, okay? But if we get sexual recombination, in other words, we get the mixing of genes by sexual reproduction instead of asexual reproduction. Number 3, okay, major factors that alter allele frequency. Number 3 has already been talked about, natural selection. Number 4, genetic drift, of which we have some types, subtypes. We have the bottleneck effect. We have the founder effect. We're going to talk about these with lab pretty soon. And then number 5, gene flow. We already talked about the fact that what natural selection is. So certain alleles get passed on to the next generation in greater proportions. A deer that's brown blends in with its environment better than a white deer. So it's going to pass on the deer that's brown is going to mate more often than the white deer, probably live longer, and pass on its genes more often. Genetic drift. There's a couple ways to look at this. One is called the bottleneck effect, a sudden change in the environment that may drastically reduce the size of the population. So, for example, in our population out here, we have a whole bunch of, let's say, rabbits, and all of a sudden we have a forest fire. And in our previous population, we had maybe 1% of the rabbits were white or albino, or let's say 0.1% of the rabbits were albino. I'll just use albino because that's an easy thing to think about. But after the forest fire, only 10 rabbits were left, and 2 of them were white, were albino. Now 20% of our population is albino instead of 0.1%, so you can see that it'd be much greater chance of passing on the white gene to their offspring, or the white allele. And it's always pictured with a bottleneck like this. Here's the original population, a few survive, and move on. The founder effect is when a few individuals become isolated from a large population. This would be like 2 birds land on a desert island, or 2 humans land on a desert island. Okay, so if there's a plane crash, like in the movie Castaway, and 2 humans end up, except in that case it was 1, but let's say 2 humans end up alone on a desert island. Okay, and so they find a way to live, and they'll have to be male and female. They survive and reproduce. Their kids are going to look more like them. That's a founder effect. If they were both red-haired people, probably all the kids living on that island are going to be red-haired. Okay, they might have 1 or 2 that aren't, but eventually that island will have a much greater population of red-haired people than normal. That's the founder effect. The 5th one we talked about was gene flow. Okay, genetic additions or subtractions from a population. This would be things like immigration and emigration. Okay, people that, or things, people, I always use people, but we could talk about anything that leaves the population or comes back to the population, changing the amount of alleles. So the primary mechanism of change, though, is natural selection, because what makes something move in or move out generally is in favorable environment. Why does somebody move to Coopersville? Why would somebody move to Coopersville? They like it here. Okay? They move here for a reason. That would be migration. Okay? But natural selection accumulates favorable genotypes. Okay? It's not necessarily favorable for this butterfly to be black. Okay, but notice that matte butterflies that emerge in the spring are orange and brown. If they emerge in the late summer, they're black and white. Okay, here they're still blending in with the leaves a little better that are laying on the surface of the forest. There they don't need to. We can have variation within a population. Okay, we can have what's called polymorphism, where poly means many, morphism means forms. This is a slide that looks at variation between two different populations of mice that live on the same island. Okay, they have chromosomal differences. Even though they look very much the same, there are differences that we can see when we start looking at their chromosomes and their genes specifically. There's geographic variation along a cline. Okay, a cline is, for example, if we go up a mountain, we have a cline. Okay, and then the higher you go, the smaller the yarrow plants can grow. So when we're up here at 2,800 meters, the tallest the yarrow plant can get is this tall. And you can talk about reasons for that and everything else, but that's called a cline. This variation is due to altitude, not necessarily due to differences in genetics. All right, so what about natural selection? Well, it increases the frequencies of certain genotypes, fitting organisms to their environment over generations. So here we go again with a list, 3 basic modes of selection. Natural selection, there could be directional selection, favoring going in one direction. It's better to be bigger. It's better to be smaller. There could be disruptive selection. There could be stabilizing selection. We can look at a graph of each of these in a minute. Okay, if we look at frequencies of individuals, here's color variations in a mouse. Okay, in an evolved population, we could have directional selection. It's better to be darker. Okay, maybe the mouse lives in Alaska, so it has disruptive selection. It's about as good to be light as it is to be dark, depending on how much snow is on the ground. Or we could have stabilizing selection, in which the middle is selected for. Okay, we get the bell curve. Okay, where whatever that perfect one is, we get a squeeze in towards the mean. We get less dark and less light. That's called stabilizing selection. The last section of this lecture is on how does genetic variation stay around. Why don't we all look the same? After all these years of natural selection, why don't all rabbits look the same? Why don't all people look the same? Why don't all fish look the same? Why don't all bluegills look exactly the same? Why is there still variation? Okay, and the first reason is diploidy. Whoops. Reason number 1 for that is diploidy. Because remember, if you have big A, little a, you're still dominant. Okay? These are, quote, hidden recessive alleles, if it's a recessive allele that causes some problem. Okay, so the fact that each parent has 2 copies of each gene is one reason. The second reason is what's called balancing selection, when natural selection maintains stable frequencies of 2 or more forms. Okay, so they kind of compete against each other. The third reason is heterozygote advantage. This is really interesting. Okay, there are many more things like this than we previously thought. And sometimes, and maybe you know this, that your dog that's a mutt dog tends to live better, longer, and better than a purebred dog. Why? Well, one of the theories is heterozygote advantage, that you have a greater advantage to be heterozygous. We're going to talk about this in a little bit of detail in lab, but the best example is that of sickle cell allele. Okay, in sickle cell anemia, it's called sickle cell anemia is the disease. Somebody that has two little a's, so if you are a little a, well, a normal blood cell, a normal red blood cell looks like this. It's nice and round and fits through the blood vessels just nicely. A little a is a hemoglobin problem that causes the blood cell to look like this, like a sickle. And so obviously it won't roll through the blood vessels very well, and they have real problems with this. But the interesting thing is that there's a disease that kills more people in the world than any other called malaria. Malaria is a parasite that infects red blood cells. Well, if you are this, your red blood cells are normal, but for some reason, the malarial parasite cannot penetrate that red blood cell as well. So, a person who is homozygous dominant, because this kills you. I mean, this is a serious disease. It can lead to serious problems and shrink your lifespan by a lot. This, you would think, would go away, because if everybody that has this is dying or not producing babies, how can we still have that allele around? Well, because being this, you get malaria. So if you look at the frequencies of the sickle cell allele, in areas where malaria, and you see the distribution of malaria caused by the protist, okay, it's not true everywhere, but where malaria is a very serious problem, many times the frequency of the sickle cell allele is really high, which I think is extraordinarily interesting and has a lot of discussion behind it for a lot of other things. For example, why American population, 50% of us are double recessive for blood type. O. Okay, is there an advantage having the O allele and other things? So then a couple other terms, neutral variation, if you read that, is genetic variation that doesn't give an advantage. Hair color in humans, for example. Sexual selection is a big deal, because here is where this is the big deal, because now this is how natural selection might work in everything. Because if you think about it, why do things look the way they do? Why does a peacock male have bright feathers? Well, because peacock females like it. Okay? So why do we have what we call sexual dimorphism, where differences between the sexes, okay? Humans have sexual dimorphism. Ducks, male ducks have sexual dimorphism. The male has a bright green head, pretty body, and the female's brown. Well, because natural selection for mating success. Okay? The better you look to another female, the better a male looks to a female, better a male peacock looks to a female peacock, he is going to pass on his genes more often. And we can talk about this in a little bit in class. So a brief discussion of why we don't have perfect organisms. Well, we're limited by the history, by how everything got started. Adaptations are often compromises. In other words, like sickle cell anemia, chance gets in the way, and selection can only edit existing things. You can't make something brand new. That's our discussion of population genetics and natural selection.